So I'd like to invite our next speaker, Dr. Epshita Islam, who is coming to us from the University of Toronto. She's a research associate who is working on preclinical development of novel vaccine formulation for Neisseria gonorrhea, among others.
Just give me 1 second. Let me try to figure this out. Awesome. Thank you so much, and good afternoon to everyone. It's a pleasure to be here to talk a little bit about gonorrhea, AMR development in this bacterium, and our efforts to develop vaccines to counter it. So gonorrhea is caused by a gram-negative bacterium called Neisseria gonorrhea. And gonorrhea as an STI, it primarily affects the genital tract with untreated infections causing complications such as permanent tissue damage of the upper reproductive tract, chronic pain, infertility, ectopic pregnancies. Besides the genital tract, it can also infect other tissues. So for instance, pharyngeal infections are rather common. And then in rare cases also disseminate into the bloodstream to cause bacteremia, as well as go into distal sites such as joints to cause arthritis, and also other complications such as endocarditis and meningitis. So what's really staggering is that the rate of gonococcal infection is very high. So in 2020, the World Health Organization estimated that there's over 80 million new infections of gonorrhea worldwide. So you can imagine that with antimicrobial resistance, if we start seeing treatment failures, it can have widespread effects on not only just reproductive health, but also on gonococcal-related complications. And another very important aspect of gonococcal infections is that prior infections do not lead to protection. So the same person can get reinfected over and over and over and over again with even the same strain. Therefore, we really need to have effective countermeasures to be able to treat this infection and or prevent this infection. So how serious is AMR development in gonorrhea? Well, it is really worrisome. So this is a table over here showing that the different classes of antibiotics that have been used to treat gonorrhea since the 1940s. So that's since the beginning of the antibiotic era. And you can see in this table the bars representing the year that an antibiotic was introduced to the year that the resistance to that class of antibiotic was first reported. And the numbers inside the graphs represent the number of years between these two events. So you can see that for some classes of antibiotics, the resistance had developed within even 5 years of introduction. And over time, gonorrhea has developed resistance towards every class of antibiotic that's been used for its treatment. And right now, we're in the last line of antibiotic regimen. And this graph ends in 2010. So this is like 15 years almost from now. And since then, there's been a rise in multi-drug resistant, as well as extensively drug-resistant strains that are now resistant to either one or both of the dual antibiotic regimen that's recommended for its treatment. So clearly, there is a need for countermeasures, and we need those countermeasures to be developed fast. So what are they? At the moment, there are some short-term options for treatment. So there is a new class of antibiotic called zoliflodacin that is in phase 3. But also at the same time, there has been more and more traction on the use of a preventative, like a vaccine, to curb AMR in gonorrhea. So for instance, there is a licensed meningococcal vaccine that contains outer membrane vesicles from Neisseria meningitidis, which is a related pathogen. And that vaccine in 2017, in a landmark paper, was found to provide some protection against gonococcal infections. So the efficacy from these real-world studies were about 30 to 40%, And this is very exciting because this is the first time that we're able to see some protection against this bacterium, and really gives rise to the possibility that if we can design a vaccine that's specifically targeting gonococcal antigens, we'd be able to see better efficacy, and it would be a better preventative option. And actually what's in last year in the UK, the Joint Vaccine and Joint Committee on Vaccine Immunizations had advised that the Group B meningococcal vaccine is recommending that the Group B meningococcal vaccine being used in high-risk populations to curb gonococcal rates. So vaccination is definitely acceptable, like, way forward. So before I jump into our vaccine development efforts, I just wanted to have the slide over here just to give a bit of more perspective on gonococcal disease. So Neisseria gonorrhea was first isolated in 1879, and within less than 100 years of antibiotic use, now it's reached the status of an urgent threat because it was able to evolve resistance to all the classes of antibiotics. However, gonorrhea is a very ancient disease. The term gonorrhea was first coined in 200 AD. Gonorrhea literally stands for the flow of seeds, referring to acute urethritis symptoms. And what's even more mind-blowing is that the earliest records of gonorrhea-like diseases are from 1500 BCE. And these are some of the earliest records of humankind. So this bacteria and this disease has been around for hundreds and hundreds of years and has co-evolved with the human host. And one thing it's done really, really well all these years is to evade the immune system of the host. And thus now we get 80 million infections every year. So this is kind of important to keep in mind as we are trying to develop new vaccines and any new therapies against gonorrhea. So typically when we are looking for, when we're thinking of developing a vaccine against gonorrhea or any other bacterium, we want to target proteins or components that are on the surface of the bacterium. Now, gonorrhea lacks a polysaccharide capsule, so the targets that we're looking at are protein targets. However, it can be really tricky to pick a good gonococcal target. So there are some proteins that are on the surface of gonococci that can actively suppress or divert a protective immune response. So we definitely do not want to pick those. And then there are other proteins that are on the surface of gonococci that exhibit antigenic variation and or phase variation. So antigenic variation, So antigenic variation basically is represented in the schematic over here, where within a population you can have the same protein, different variants of the same protein. And phase variation is shown over here in the green, where the bacteria can turn off and on the expression of proteins with replication cycles. So both of these types of variations make it difficult to provide broad coverage with a vaccine, and also it's tricky to make sure that the bacteria is not just turning off a receptor or like a protein to escape the vaccine pressure. And then the third aspect for developing gonococcal or any vaccine is practicality. So there are vaccine candidates that may look great, but can just be very difficult to produce on a commercial scale. So if we want to think about what would constitute an ideal vaccine candidate, here are some criteria that we typically look at. So we want the antigens to be present on all strains, and we ideally want the antigen to exhibit minimal antigenic variation or be conserved in order to provide broad coverage of all strains. Ideally, a vaccine candidate would be essential, so it should not just be, go through mutation or loss or just turned off due to vaccine pressure. And then we ideally need this antigen to be scalable for commercial manufacturing. So this brings me to a very promising vaccine target in Neisseria gonorrhea called the transferrin binding protein B. So transferrin binding protein B is the part of the gonococcal transferrin receptor complex. So the way that transferrin binding protein B works is that it is a surface lipoprotein, so it's found on the surface, on the outer membrane of Neisseria gonorrhea, where it's anchored via a lipid anchor, and then it extends out, and it's able to grab human transferrin. So human transferrin is an iron carrier protein that is abundant in blood, and other mucosal secretions and bodily fluids. And the way so TBPB grabs onto human transferrin and brings it to the other component of the receptor system called TBPA, and then the iron is taken into the bacterium and HGF is released. So we think that TBPB is a really great vaccine antigen because it satisfies most of the criteria that we have for an ideal vaccine antigen. So we know that the soluble part of TBPB can be produced very readily and can be scaled up. The transferrin receptor system is present on all strains, and we know that it's essential because iron is an essential micronutrient, and this transferrin receptor system is able to, it serves that essential function in acquiring iron. However, we're not that lucky, so unfortunately, based on our understanding of TBPB from the literature, we knew that this was not a conserved protein. And so that was something that we would have had to deal with for providing vaccine coverage. So we wanted to get an idea of what's the overall diversity of TBPB protein. So this was work that was done by Dr. David Curran, who's a really great bioinformatician in our group, who basically pulled out all the publicly available genomes of Neisseria gonorrhea to construct a TBPB tree. Okay, so this tree, this is the current tree. It has over 1,200 different TBPB sequences. And that's a lot of different sequences and a lot of different variants that are present there. However, when he kind of clustered them, they really form into two different clusters. So that's a bit more manageable than trying to just look at 1,200 different sequences. However, just looking at this tree, now we're posed with the challenge that, like, okay, if you want to pick an antigen from all these available variants, what would we pick, right? So what Dave ended up doing was he designed a custom software which is able to take the phylogenetic distances of all the different TBPBs and provide what is the centralmost variant. So those are indicated over here by TBPB1 and TBPB2. So these are the most representative variants within each of the clusters. And we hypothesized that by immunizing with the centralmost variant, we should be able to elicit a broad immune response that can detect all variants within the cluster. And this was our way to rationally select these variants instead of just doing it by eye, which is you're just leaving it to luck at that point. So we wanted to rationally select our vaccine antigens. So the way that we tested this was by immunizing rabbits with either TBPB1, TBPB2, a combination of TBPB1 and TBPB2 in a bivalent composition, and TBPB1, TBPB2, plus 3 additional TBPB antigens in a pentavalent composition. So over here, the heat map is showing the reactivity of antisera after 1, 2, and 3 doses of each of these vaccines. And on the rows of the heat map represent different TBPB variants that were picked from different parts of the phylogenetic tree. So we wanted to see how well the antiserum is able to cover or react to these variants. So in the dark blue, the darker the color, it means more reactivity. You can see that with one of the 2 proteins, either TBPB1 or TBPB2, you get a reactivity against a subset of the TBPB. However, when you combine those two in the bivalent formulation, you get reactivity against all the TBPBs that we tested in our panel. And addition of 3 additional antigens don't really improve it. So we were very confident that based on this, that a bivalent composition would be able to provide at least antibody coverage. But we obviously wanted to follow this up with some more rigorous preclinical work. So we took these antigens and we tested them in a mouse model of gonococcal infection. So this mouse model is kind of the gold standard that's used for vaccine evaluation. In this model, you take female mice that have been treated with antibiotics and hormones to render them susceptible to gonococcal infection in the lower GI tract. And then you infect them and monitor for how long they are colonized. So if a vaccine is successful, you can see that the mice are able to clear gonococci faster than the control group. So that's what we're seeing over here. When we challenge these mice with either a cluster 1 or a cluster 2 strain, we do see accelerated clearance. So now we've shown in 2 different models that this bivalent composition is working. So at this point, we were very, very optimistic that okay, we have a bivalent composition. We successfully managed to cover the diversity of this target. And we started thinking ahead of okay, what's going to be the next steps for clinical translation? And what we realized at this point was that our preclinical models did not take into account one very critical interaction that would be present in humans. So if you remember, TBPB binds to human transferrin, and this binding is very specific to human transferrin only. So in our rabbit or mouse studies with wild-type animals, we do not have this interaction happening. So what we started thinking is okay, what will happen, let's say if you have TBPB vaccine and you inject that into the arm. And we worry that as soon as you inject the vaccine, transferrin, which is very abundant in the body, it's found in milligram quantities in serum, would come into contact with your vaccine antigen and complex with it. So this could be problematic for 2 different reasons. First, that means that if 30% of the vaccine's surface is covered by human transferrin, you would not get a optimal immune response. And the second concern was that if HDF is stuck to your vaccine antigen, what if you start getting some response against human transferrin itself, like autoantibodies? So we really needed to figure out a way to overcome this challenge. And what we thought would be the best approach would be to surface modify our TBPB antigen in a way that abrogates this interaction with HDF. So that's what we set out to do, and this is work done by Natalie, who is a PhD student. And what she had done was she took TBPB1 and TBPB2 and made specific mutants of these variants that are able to abolish HDF interaction. So this is just looking at a comparison of wild-type TBPB versus mutant TBPB. So the difference between the wild-type and the mutant is in a single amino acid residue. This is looking down at the transferrin binding interface, and this is a charge map. So what the mutation does is it alters the charge on the transferrin binding interface. Other than that point mutation, the mutant TBPB antigen behaves just the same way as the wild-type TBPB antigen, in the sense that with all the biophysical tests that we've done, they act identically. So we think that the mutant and the wild-type are structurally identical to each other. So over here, you have the thermal profiles that just look identical. However, functionally, the mutant antigen is now no longer able to bind human transferrin. So that's shown in the dot plot over here, where with the wild-type TBPB, you get a signal when you probe with human transferrin, but with the mutant TBPB, you get no signal at all. So we've successfully been able to abolish the interaction between TBPB and HDF, and we think that this would result in a better antigen. However, we still needed a way to test it. And the way that we tested it was by utilizing a mouse model. So this mouse model is a transgenic mouse that expresses physiological levels of human transferrin. So what happens when we start doing a similar colonization experiment in this mouse? So in this mouse, human transferrin, just as in humans, is present during the development of the immune response. So when you challenge this mouse, what we find is that the mutant antigen is able to provide accelerated clearance, that is shown in the red, whereas the wild-type TBPB and the adjuvant don't look any different anymore. So we really think with all the evidence that we have with the mutant antigen that this is going to be our go-forward antigen for this vaccine. So at the same time that we're thinking of developing our antigen composition, we were also looking at other components that go into a vaccine. So protein-based vaccines typically need an adjuvant because proteins are typically very weakly immunogenic on their own. And the vaccine adjuvants, now there are a wide variety of adjuvants that are available, and they can have different physical properties. So some adjuvants are emulsion-based, or they can also have different immunological properties. So some adjuvants can have different mechanisms of action. So for instance, they can form like a depot with the antigen and allow the antigen to persist for longer. They can also help with antigen uptake by antigen-presenting cells. Adjuvants, depending on like what components these have, can really modulate the immune microenvironment at the site of injection. And this can have downstream effects on the type of immune response that develops after vaccination. So to test what type of vaccine adjuvants would be optimal for gonococcal protection with TBPB, we tested 5 different research-grade adjuvants at this point. So these included 3 emulsions that are shown at the top panel and 2 alum-based formulations that are shown at the bottom. And what we found from these colonization experiments was that for all the different formulations, there was a reduction in the median days colonized by the vaccinated groups. However, the best separation between the vaccine and the control groups was seen for both the TH balanced adjuvants. So the TH balance basically refers to the type of T helper cell response. So TH2, TH1, or TH2 skewing, TH1 skewing, or TH1 to balance basically refers to the type of helper T cell response, and that can have different downstream effector functions. So we found that the balanced adjuvants performed the best in our hands. So at this point, we have a really good idea and kind of all the pieces that we need to come up with the final formulation. So we know that for our TBPB-based vaccine, we would need 2 non-binding TBPB mutant antigens with an adjuvant, and most likely this would be a TH-balanced adjuvant. And we expect that this vaccine would be able to cover all the different gonococcal strains. However, knowing how easily and how effectively like gonorrhea is able to evade any countermeasure that you throw at it, we can't just stop here. So we are also at the same time like looking at other potential antigens and trying to figure out ways that we can develop effective vaccines, vaccine antigens that target other potential systems. So over here, I'm showing the outer membrane of gonococci, and these are a bunch of different other receptor complexes that are needed for nutrient uptake. And these are ones that some other potential antigens that we're looking at developing at this moment. Most of these satisfy the the requirements of an ideal antigen in that they're essential, present on all strains. However, again, like some of these antigens are integral membrane proteins, so they're very difficult to produce. So we're looking at ways that we can develop hybrid antigens or different production methods to be able to overcome those limitations. And then we are not the only group that's working on gonococcal vaccines. Other groups are working on it as well. There are groups that are working on gonococcal OMVs or outer membrane vesicle-based vaccines. That's similar to the group B meningococcal vaccine with which you saw cross-protection. There's other groups that's looking at targeting gonococcal lipooligosaccharide, as well as other proteins that are involved in adhesion and efflux. And then I didn't list it over here, but then there's also a lot of proteomic and reverse vaccinology studies also occurring at this moment, trying to identify new candidates. So just future outlook, I think that it's not a matter of if, but really a matter of when we come up with some really great vaccine options against gonorrhea. So with that, I would like to move on to acknowledgements. I would like to thank Scott Gray-Owen and Trevor Moraes, whose labs I work in. Anthony Shrivers, he is the person who actually discovered the transferrin receptor complex about 30 years ago, and he's a great scientist to have on our team. And then Dr. Cindy Cornelison did some of the foundational work that showed that the transferrin receptor is essential, and she is a close collaborator in all of our vaccine-related projects. And of course, thanks to the big team of people involved in gonococcal vaccine projects. Special shout out to Dr. Jamie Fagan, who did a lot of the immunological studies that I've shown today. And with that, I would like to end, and I'd be happy to answer any questions.
So thank you. We have a few minutes for questions.
Hey, Epshita. That's a very nice talk. That was very exciting, and I'm just curious. Can you comment on the applicability in general of vaccination against AMR bugs? Yeah.
So, I mean, in terms of gonorrhea, for instance, like, we do functional assays and try to test our vaccines against strains, including AMR strains. And like our vaccine targets are not targeting systems that are involved in AMR, but it's general. So like, we expect that AMR strains to be covered as well as non-AMR strains. And I think like, it's just a general strategy. I think that could be used for not just gonorrhea, but for any AMR pathogen. If you can reduce the rate of infection, you reduce the antibiotic consumption. And I think that has the downstream effect of just reduced resistance developing overall in the population.
Yes, I see that. And I'm just mainly wondering, so gonococci is a pretty hard bug to vaccinate against, and it seems like there's good progress on that now. And so with all of our other pathogens, bacterial pathogens that are AMR, have problems due to AMR, do you have any thoughts on whether vaccination would be a good strategy for those as well, or is there something specific to gonococci that makes it amenable for this?
No, I think vaccination would be a broad strategy for any kind of infection.
Cool.
Yeah,
Thanks.
and I think yesterday we heard that like the concept that like maybe even vaccinating against viral pathogens can have an improvement of AMR outcomes, because if you can reduce the disease symptoms and number of patients who go into the ER, like we reduce like the number of prescriptions that are handed out for antibiotics. So, vaccination in general can be a very effective way to curb AMR.
Yeah, that's really cool. Thanks.
Thanks, Freeman.
That was a really nice talk. I'm curious, in the animals that remain colonized, right? So the animals for which you don't see protection, have you looked at those strains, and do you see any evidence of mutation in the direction of vaccine avoidance or anything like that?
So we haven't tested specifically what strains, like, what comes out of the bacteria, and whether or not those have like turned off the receptor or not. That's a really good concept. What we have tried to look at is like differences in the immune response between like the mice that are that clear infection fast versus remain colonized, and we haven't really been able to find like a good correlate yet. But for sure, that's a really great idea that like just looking at like what's coming out would be very informative to see if we're seeing drifts in the bacterial population to avoid the vaccine.
Any last questions?
Hi, thank you. That was really interesting. I was wondering what kind of the end goal with the vaccine is. Is this a preventative vaccine for a general population, or maybe a population that's more at risk, or maybe more for people who have had infection to prevent recurring infection?
Absolutely, and I think it's a very, very broad question, really, about like general like uptake of a STI vaccine. So, for instance, in the UK, for instance, like the meningococcal vaccine is specifically being recommended for target populations. So, if there's a priority, perhaps like a target population would be the first population to utilize the vaccine against. But in general, I think with education, we can hopefully get a broader vaccination of the general population of adults and right before like sexual activity. So I think that would have like the broadest impact.
Thank you so much.
All right. Well, thank you very much.